So in this video we're going to see the urinary system anatomy. So the kidneys are located within the abdominal cavity. They are retroperitoneal organs and they are paired, and they have this kidney shape, this pinto bean shape that it has this convexity that faces laterally and this concavity that faces medially. Now this concavity is known as the hilum, and the kidneys are protected by a membrane that's called the renal capsule. And then on the superior pole of the kidneys, which is this, we have the adrenal glands. Now the kidneys are supplied by the renal arteries, which are branches of the abdominal aorta, which is this artery. Now the abdominal aorta is located towards the left side of the spinal column. And then this is the inferior vena cava that drains the blood from the kidneys, and these will be the renal veins. And the inferior vena cava, it is located on the right side of the aorta. So if you see here the difference in length between the renal veins. So this is the left renal vein, this is the right renal vein. So this is the right kidney, this is the left kidney. So the left renal vein is longer than the right renal vein because the inferior vena cava, it is located towards the right. And it is the same for the same kind of relationship at some point of the length of the renal artery. So since the aorta is located towards the left, you expect that the left renal artery is shorter than the left renal artery. So let's see how it shows in here. So this is the left kidney, this is the right kidney, and if you see, this is the left renal artery, this is longer than the right. Okay, so that's one thing to keep in mind. Now, this is the actual kidney model that shows better the details of the kidney. So in this model, you can see again the pinto bean shape of the kidneys, and this capsule that protects directly the kidney is called the renal capsule. Now outside of the capsule, we have a fat pad that we call the perirenal fat, and then outside of the perirenal fat we have another membrane that protects the kidneys that is called the renal fascia. Now just next to the capsule we have this major region that is called the renal cortex, and then in here, just medial to this or deep to this cortex, we have the renal medulla, and within the medulla we have different structures. So we have the renal pyramids, which are this. So the pyramids, they have a triangular shape with the base facing towards the cortex and the apex facing towards this indentation, which is the hilum. So these are the pyramids, and then in between the pyramids we have the renal columns. The renal columns allow the entry and exit of the blood vessels. So these are the blood vessels that goes into different regions of the kidney, and also we have lymphatics and nerves running into these renal columns. Now within the hilum we have different structures. So we have the renal artery, which is this. Here is the renal vein, and then this will be the renal pelvis, and outside of this hilum we have the ureter. The ureter brings the urine from the kidneys into the urinary bladder, and then at the tip of the renal pyramids we have minor calyces, which are this. The minor calyces, 2 to 3, join to form a major calyx, which is this. And then 2 to 3 major calyces, they form this funnel-like structure that it is called the renal pelvis. Now, within the medulla, and inside the pyramids, we have the collecting ducts. So this is where the urine it is drained, and also we have loops of Henle, which are this. And within the loop of Henle, we have different filtration, but we will see this in the next part of the model. So this is part A. And then there is this segment of the kidney that we call the lobe. The lobe is formed by a single renal pyramid and its corresponding part of the cortex. So if we magnify this renal lobe, we will be able to see the different structures. So within the cortex of this renal lobe, we have two different types of nephrons, which are the structural and functional unit of the kidneys. So we have the cortical nephrons that are closer to the capsule, and then we have the juxtamedullary nephrons that are located closer to the medulla. So now, how can you tell which ones are the cortical and which ones are the juxtamedullary nephrons? Well, by the length of this part that is called the loop of Henle. So the loop of Henle, it is longer in the juxtamedullary nephrons. Why they're called juxtamedullary nephrons? Because they're closer to the medulla, which is this part. Juxta means nearby, and then medullary, next to the medulla. And then these will be the cortical nephrons because they have a short loop of Henle. Now, another way to tell which one is which one is cortical, which one is juxtamedullary, is by looking at the blood vessels that are closer to the loop of Henle. So if the blood vessels run straight parallel to the loop of Henle without making a U-shaped connection, this will be the vasa recta. And if the blood vessels, they form like little connections in a U-shaped like form, this will be the peritubular capillaries. Now, then the nephron, it is formed by different parts. The first part that is called the renal corpuscle, which is this, then the renal tubules, which are all of these together, and then the collecting duct in here. Now, the renal tubules, if they're folded like this, and they are close to the renal corpuscle, this will be called proximal convoluted tubule. And then you have a loop with its descending limb, the loop itself, and the ascending limb. And then you have another tube that it is folded that is called the distal convoluted tubule. Remember, proximal means close by, distal means away. So this is the proximal convoluted tubule, or PCT, and this is the distal convoluted tubule, or DCT. And in between the DCT and the PCT, you have the loop of Henle. And then you will have the collecting duct in here. Now, these blood vessels that you see here are branches of the renal artery, which is this. And when this renal artery enters into the hilum, it's going to divide into 2 to 3 segmental arteries, which are this. These are the segmental. And then the segmentals, once they enter into the columns, are going to be called interlobar. And then the interlobars, they're going to make this turn towards the pyramids like this, and these are going to be called arcuate. So these ones that you see here are the arcuate blood vessels. And then the arcuate, they send these straight branches towards the cortex that are called the cortical radiate, which are this. And then from the cortical radiate, you have one arteriole that goes and forms a net of capillaries in here, which is called the glomerulus. But this arteriole will be called the afferent arteriole. And then you have exiting from the renal corpuscle another arteriole that is called the efferent arteriole. So the arriving arteriole is going to be called the afferent or arriving, and then it will form a tangle or a tuft of capillaries that we call the glomerulus, and then we have an exiting arteriole or efferent, and then you will have the blood supply going into these peritubular capillaries or into the vasa recta, depending whether it's a cortical nephron or a juxtamedullary nephron. And then from there, the blood will go into these cortical radiate veins and then into the arcuate. And then from the arcuate, it will go into the interlobars, and then from the interlobars into the renal vein. So there is no segmental veins. So let's go again over the circulation then for the kidney. So from renal artery into segmental artery, into interlobar artery, into arcuate artery, into cortical radiate artery, and then into the afferent arteriole, and then into the glomerulus, and then into the efferent arteriole, and then into the peritubular capillaries, and then into the arcuate vein, and then into the interlobar veins, and then into the renal vein, and then into the inferior vena cava. So that's how you have the renal blood circulation. Now, if you are producing urine, you will have pressure building up into the renal corpuscle. So let's see how this happens. So this is a magnification of this region, the renal corpuscle. So in the renal corpuscle, we have an outer structure that it is called the renal capsule, that it is made of simple squamous epithelium. And then you have this afferent arteriole or arriving arteriole, and then you have this efferent arteriole. If you see the diameter of the afferent and the efferent arteriole, this is much wider than the efferent arteriole. And this knot of capillaries that you see, this will be the glomerulus. So as the blood is entering into here, it's becoming filtrated. Why? Because you build up pressure by having a lower or a less diameter here in the afferent. So since you have more arriving blood than what is exiting, you have built up pressure, and then in here in these capillaries that has fenestrations, the substances exits and they form a fluid that we call the filtrate. And this filtrate is going to be rich in ions, nutrients, and then the filtrate is going to move into this space that we call the capsular space. Now, another name for the capsule of the renal corpuscle, it is the Bowman's capsule. Just like almost the car dealership here in town, but no, it's Bowman's capsule. And then this glomerulus, it is actually surrounded by these cells that are called the podocytes. It's fit-like cells that has finger-like projections that you see here that are called the pedicels. So these pedicels open and close to allow the passage of different substances into the filtrate. And the substances that it can go into here, it can be anything with the exception of proteins and red blood cells. So those are two things that shouldn't go normally into the filtrate. Now the filtrate will pass then from the capsular space into the PCT, into here, and you will start absorbing from this filtrate that now after it goes into the PCT, now it's going to be called the tubular fluid. You start absorbing water, sodium, amino acid, glucose, whatever substances your body is going to reclaim. Now the epithelium of this PCT or proximal convoluted tubule is simple cuboidal with microvilli that allows the absorption of these substances. Now in here we have, just next to this afferent arteriole, we have the DCT, and then we have these cells, which are the macula densa cells, And together, all of this structure in here, you form a structure that is called the juxtaglomerular cells or juxtaglomerular apparatus. This juxtaglomerular apparatus is important to detect changes in blood pressure, so you can start a cascade or a mechanism that is going to control the blood pressure. So if you have low blood pressure, for instance, you can secrete renin, and this renin is going to be an enzyme that is going to break down a protein or a polypeptide that we have in our blood that is called angiotensinogen. Angiotensinogen, with the action of renin, is going to be broken down into a smaller peptide that is going to be called angiotensin 1. And angiotensin 1 is going to go into the bloodstream and then is going to reach the lungs. And in the lungs, we have an enzyme that is called the angiotensin converting enzyme. So it's going to convert angiotensin 1 into angiotensin 2. And angiotensin 2 is going to constrict your arterioles in your body so you can increase the blood pressure. And not only that, but this angiotensin 2 is going to stimulate the secretion of aldosterone by your adrenal gland so you can increase the absorption of water and sodium in your kidneys so you can increase blood volume and thus increase blood pressure. So then in here, in the renal corpuscle, you form a filtrate. The filtrate will move into the renal tubules, and now it's going to be called tubular fluid. So in here in the PCT, you absorb the majority of the water that goes into the tubular fluid and the important nutrients and ions that your body requires. So you reabsorb in here. And then as you move the tubular fluid through the loop of Henle, you have variable reabsorption of water and electrolytes. And then finally, you reach into the DCT, and in here you have still absorption of water, but in this case it's going to be under the influence of aldosterone, and actually you also have absorption of sodium, and then you secrete drugs and toxins within this region, and then the fluid will pass from the DCT into the collecting duct, and in here in the collecting duct you can still absorb water, and then you secrete ions like hydrogen, and then you can control the pH of the body. And also you have here within these structures internally cells that are capable of balancing our pH, and you can concentrate the urine in the medulla by having variable amounts of sodium chloride outside of this region in the medulla so that the water can move across from the tubules, from the loop of Henle into the vasa recta. So one of the functions of these juxtamedullary nephrons is to concentrate urine besides producing urine, which is mainly exclusive function of these cortical nephrons. So this is all for this model and in this part of the urinary system. So I will see you in the next video.